Good morning, everyone. My name is Lisa Mulcahy, and I'm with the Smithsonian Marine Ecosystems Exhibit. And I'm very excited and proud to introduce Dr. Jessica Zehnpfennig. She is a postdoc at the Smithsonian Marine Station, and I'll pass it over to her. Thanks for joining us, and we have two more yet to go, so be looking for the schedule.
Thanks, Lisa. So today I'm going to be talking a little bit about some of the research I've been doing as part of the Smithsonian's Ocean DNA Initiative. And what I'm focusing on is bridging our genetic gap and analyzing what eDNA markers are available for benthic and faunal species in coastal lagoons. So what does that mean? We'll get into that, but I just want to give everybody a little bit of an about me slide. So my name is Jessica Zehnpfennig, as Lisa already introduced, and I actually attended Central Michigan University almost without stop from 2012 to 2024. So I started in 2012 for my bachelor's, and I graduated in 2017 with my bachelor's of science and biology with a concentration in natural resources. And then I took like 7-month break where I did animal control for a little bit. And then I went back to school for my master's in 2018, and I got that in 2020 where I looked at environmental microbiology. So I analyzed microbial community transcriptomes. So I looked at microbial community gene expression in the Atlantic Ocean, and that led me to love marine science and love the marine systems a little bit more. And I started my PhD in 2020, and where I earned it in 2024, where I got my doctorate in ecosystem sciences, and my thesis was biodiversity and evolution in molecular ecology of Antarctic sea spiders. So I've had a nice little speckled past in science, and that led me to my current position as a postdoctoral research fellow at the Smithsonian Marine Station here in Warpace. Those are my dogs, Koda and Toby. They're with me everywhere I go. But let's get down to business. Before I wanted to give you a whole overview of what we're doing, I wanted to explain some things for anybody who might not know exactly some of these terms. So we study benthic and faunal, and benthic really means bottom of the marine floor. So benthic comes from benthos, which is a term that was used to describe the bottom. And fauna are the organisms that live within the sediments of the seafloor. So they burrow and they live kind of beneath the surface a lot of the time. These are the infauna. Epifauna are organisms that live above the seafloor, but they're still on the bottom. So you think of like a sea star would be more epifaunal. And then one last term I used SERP a few times, and SERP stands for Comprehensive Everglades Restoration Project. But my talk's about eDNA, or environmental DNA, so I wanted to give everybody an overview of what environmental DNA is. Environmental DNA originates from when organisms in the environment shed their cellular materials, so they will skin cells, feces, saliva, anything that can come from a person, it's shed into the environment, and it stays in that environment for a set amount of time. And what we do is we try to collect that DNA and analyze it, and we do that through metabarcoding. So in metabarcoding, we extract DNA from our environmental samples, and then we use something called polymerase chain reaction, or PCR, and that targets a barcode region, which is usually CO1 or ribosomal IPS region. So we target a specific region of the DNA, and we were able to do that through PCR. We put that amplified region through sequencing, and then through bioinformatic pipelines, I'm able to identify amplified sequence variants, or ASVs. Those are like individual sequence variants in there, and those are what tell us what our species are. And then from there, I can get a taxonomic assignment and look at diversity of an ecosystem. This is a nice little schematic I really use. We can plug that into the DNA. We can kind of tell what organisms are there through that. So I always like to say it's kind of like CSI Benthic division, because if you think about crime shows, they always say like the killer left DNA at the scene of the crime. And that's basically what we're doing in marine ecosystems, only we're not studying killers, we're studying diversity, which is still really interesting. And one of my major focuses of my research is looking at eDNA sampling to enhance monitoring in the Indian River Lagoon. And we do this with Dr. Holly Sweat's team. She is head of the Benthic Ecology Lab at the Smithsonian Marine Station. And I go out and I've been conducting quarterly samplings coupled with her comprehensive long-term restoration project benthic and faunal monitoring program. So that program's been around for over 20 years. They have a huge really wonderful data set available of what they've found. We monitor 15 sites, so we were those sites in the Indian River Lagoon, and St. Lucie estuary. And how Holly does this, really briefly, this is just a brief overview. One of the ways that she analyzes these ecosystems and does assessments of these ecosystems is through something called the M-AMBI index. The M-AMBI index stands for Multivariate Active Marine Biotic Index. And through that index, she's able to get a report card assessment of the ecosystems. So basically, if it's in great shape, great, it gets an A. If it's in kind of not so great shape, we'll give it like a C. But if it's in not great shape at all, it gets an F. So she has a really great, well, Borja all developed it, but it's a really great tool that we use to assess the ecosystems in those benthic and faunal communities. The only downside to this index is that it requires count data. So that means we have to go out, collect everything, and then go through and individually sort through and find all the species that are in the graph. That is time consuming, costly. The taxonomic identification of each individual can be difficult because sometimes during the grab or even just through preservation, they get a little degraded. So one question I'm looking at is, can we use our eDNA to really enhance monitoring efforts? Can we get the same M-AMBI index score using our sequencing reads and being able to identify the infaunal in that community through sequencing? And if we can get the same M-AMBI index values, then we can expand our monitoring. It would can allow us to go to more spots. It can also allow us to do something called rapid eDNA monitoring. So when a hurricane hits, we all know we have a decent amount of notice when a hurricane's about to hit, at least a couple of days. We could go to that spot, collect the community, and then after the hurricane hits, go back and collect from that community and see if there's a change. And that would allow us to have a more rapid response to these stressors, oil spills, anything that could stress the ecosystems out. So how do we do this? I'm gonna try here. So I have a video of our sampling.
There, I see the pointer. You got it.
So
join us as we collect field samples for our Ocean DNA initiative. This is Zehnpfennig, a postdoctoral fellow at the Smithsonian Marine Station, working at one of our demonstration sites in the Indian River Lagoon. After arriving at a site, our first step is to sanitize all our equipment in a diluted bleach solution. We also wear gloves to prevent contaminating the samples with our own DNA. After sanitizing, we rinse everything off in the water and prepare for sampling. We use two main methods to collect our samples, the ponar grab and the sediment core. For the ponar grab, we set the spring pin, lower it into the water, and tug the rope to trigger the grab. Once it's secured, we haul it back up, empty the sediment into a sieve, and collect a few spoonfuls into a sterile cup. The remaining material is sieved out and then preserved in ethanol for further genetic analysis. Next, we use the sediment core. Since it's too large to fully sterilize, we employ our core in a core method. Using a modified syringe, we extract a smaller core from the center of the main core and store it in a sterile container. Once everything is collected, we place the samples on ice to keep them fresh until we get to the lab, where we'll put them in our negative 20 degrees Celsius freezer. After putting all our supplies back away, we're ready to sample the next site. Stay tuned for part 2, where we'll show you how we process these sediment samples for genetic analysis.
The next thing I want to do is we do processing. So I'm going to get really close to that. So this is a continuation. Sabrina was awesome. She made a couple videos.
Back in October, we showed you how we collected sediment samples to be used for ocean DNA. So now, let's put on our PPE, disinfect our workstation, and show you the first part of processing those samples, decantations. First, we pull a sample from the freezer and let it thaw while we prep our supplies. We'll need a sterilized 2-liter Erlenmeyer flask, 45 micron and 2 millimeter sieves, a mortar and pestle, a spatula, and a scale. We start by transferring the sample into the flask, rinsing the container with saline water to ensure we get everything out. After sealing the flask with parafilm, we begin decantation. This involves swirling the sediment in the flask to suspend organic material, leaving heavy sediment at the bottom. After a minute of swirling, we pour the suspension through our stacked sieves. The larger mesh catches debris like leaves and sticks and any possible voucher specimen, while the smaller mesh traps the organic matter that we will extract DNA from. We repeat this process until the saline runs clear, which can take several rounds for mucky samples like this one. Next, we weigh the collected organic material, split it in half, and grind 1/2 in the mortar and pestle for a minute to homogenize it. We'll return the homogenized material to its vacuum tube, preserve it in ethanol, and freeze it along with the other half we keep as an archive sample. Finally, we disinfect all supplies in a bleach solution and get ready to decant the next sample. Stay tuned for part 3, where we'll show you how we actually extract DNA from the organic material we've prepared.
So next, I go through DNA extraction.
Back in October,
and this is super lengthy, but this is the process of DNA extraction. It includes. Oh, I'm on the mouse. Okay, I think I'll just. Okay. Anyway, so this is our DNA extraction process. So basically, I take those sediment samples, I incubate them, and then I go through a ton of processes where I add different amounts of chemicals with a Kaizen extraction kit. I just wanted to show everybody the lengthy process of this, and that it takes a while to get our final DNA concentration. But once we get those DNA concentrations, we then send them up to the NMNH in DC, where they will do any preparation and sequence and do metabarcoding sequencing for us. They get sequenced usually on an aluminum next generation sequencer. And then after I get that sequencing back, I then run it through bioinformatic pipelines, and those bioinformatic pipelines allow me to identify those individual ASVs, and then I can assign taxonomy through that. So I use something called BLAST, and BLAST stands for Basic Local Alignment Search, and it allows me it compares the DNA that I'm giving it to a reference library that's already there. So anything that's in the library that we've created, if it's a match, it'll let me know, and at what percent it'll let me know. And this is helpful because that's how I can determine what is in our system and give us answers. But let's talk a little bit about reference libraries, because that's been the main focus. So I talked about BLAST and reference libraries, but accurate species identification using eDNA. I really gotta get off this box. Anyways, accurate species reference of eDNA sequences relies on reference libraries. So I really do like to think of reference libraries almost like the library we used to go to when we were a kid. All those books, and you go and you'd be like, I really want to get this Harry Potter book, or I want to get this Stephen King book, and then the library would be like, oh, I don't have that book by Stephen King, but I do have it. And you're like, oh, but I didn't. I wanted The Shining, but it's close. It's still horror, but it's not quite the same. So I kind of love that analogy because that's kind of what I do with what's available to us in our reference libraries. If we have it, we know there's a match, it's great. I can say, yeah, I was looking for this, we have it in there. However, some reference libraries might not have everything that we're looking for, which is an issue. But there are some pros, no matter what. We're going to talk about some advantages, which they help facilitate species level identifications. So like I said, those degraded samples or those older samples or larval samples, sometimes you can't get a good species level identification with the knowledge you have. If you're not a taxonomist like me, I'm a molecular ecologist, it's hard for me to be able to identify things 100% all the time. But with these reference libraries, if they have it in there, it'll tell us, yep, this is a match. So this picture here, you can see there's like 100% match in these sequences. So that's kind of how that works. And that helps us support our biodiversity monitoring and enables us to have large scale studies. The larger your reference library, the bigger you can go, and the more you can analyze. However, like I said, some disadvantages. So identification accuracy is something that we're working on. Sometimes if it's not in the system, it won't be identified. So that's our biggest issue, is looking for things that are in our system and what's not in the system. Coverage limitations, so new species, we're always discovering new species almost every day. Sometimes they don't have the barcodes in the reference libraries yet. Cryptic species, or species that are often hard to find, are also often not in our reference libraries. Sometimes quality consistency, so like I said, I'm not a taxonomist, but sometimes people like me still will do taxonomic work, and I try to be the best I can at identifying, but sometimes people will misidentify things and put them on these public reference libraries, and with those misidentifications, it's not the right, when you hit it and hits to a good thing, you're not going to the right species. And then quality, sometimes sequences are degraded and they still get put on there, so you get a not so great sequence that in there is your only reference. And this leads to some unreliable conclusions and misidentifications. which can be problematic. But what I wanted to do and what the main research, what I've mainly been focusing on is looking at the availability of some eDNA markers for those Andes species that I talked about found in the Indian River Lagoon. So to do this, I used Holly's group's long-term monitoring data the last 20 years. I looked at all of the Benthic species that they've collected. I put it into a list, and then I used the Indian River Species Lagoon inventory as well to then go further and look for all the species that are on that inventory list as well. And I then cross-referenced that list off of the most recent Andes species list. So these Gillette et al. 2015, Boral et al. 2020, they continuously look and try to classify species on the Andes list and tell you what Andes species are there. So I then was like, okay, well, I want to see how many of these species that I created in my list are Andes species. So I cross-referenced it. And then I went to publicly available databases such as NCBI and Bulge, and I looked for these 5 common eDNA markers: CO1, 12S, 18S, 16S, 28S. And so if they were present, they got a yes, it's there. If they didn't have it, no, we're not there. And because I had 1,188 species, I sorted them into 10 major taxonomic groups. So each species belongs to a higher level taxonomic group. And that's what these are. So we looked at amphipods. So these are amphipods up in the right corner. Annelids, which are our worms. Bivalves, which are like those clams. Cnidarians. So cnidarians include hydrozoans, etc. Crustaceans, so any crabs or shrimp. Decapods, which are had a decapod right here. Here's a decapod. Echinoderms, which include our brittle stars, sea stars. Gastropods, which are these nice little swirly shell insect larvae. So we get a lot of chironomid or we get a lot of insect larvae that they'll spend their time in fresh or pretty fresh water. So we were looking for those as well as any other taxa so that were on that list. So basically, these are the major groups that I inserted into. So then I looked, and if it had at least one of the 5 available reference library, or one of those 5 available barcodes, I said yes, it's got a barcode. So this graph here just shows the number of taxa, those higher level taxonomic groups. And then the blue-green color shows yes, there's barcodes. The gray shows no barcodes available, and this is a proportion of each one. This black dotted line just shows the lowest number. So insecta, those insect larvae had the lowest number of taxa. So it's just a reference to show like there's a lot more annelids than insects. And what I found out of this is that we have some really good coverage. We have like in cnidarians, we have a high proportion of species, Andes species with at least one barcode. Decapods and echinoderms are still relatively high as well. But then other species like annelids are only a little over half. Bivalves are still pretty low, and then gastropods are under half of the species don't have a reference library available for them. And then what I wanted to do was I wanted to look at the coverage of how many barcodes across taxa. So if they had 0 through 5, so if they had none versus all 5 of the barcodes, I wanted to look at that. So that's what I made. I looked through and I found that 40% of the species on my list do not have any of the genetic markers available. And only 6.17% of them have all 5 reference library or all 5 GNA barcodes available. This is important because this is saying like, okay, so when we look at our bar graph here, if you look between 0 and 1, only having 1, you're still over half of those species only have less than 2 reference DNA barcodes in a publicly available reference library. So I was like, ooh, let's look at what barcodes are available. So these next slides, they're a little complicated, but I just was looking at each of those 10 groups, which is these lists. Once again, the blue means barcodes, the gray means about, and then that black dotted line shows the insect or like the lowest number for a reference. And each one of these taxonomic groups, I just looked at those 5 markers I talked about. So the 12S, which is from left to right, it's 12S, 16S, 18S, 20S, and CO1. And what I really was looking at is just the availability of each one of those markers for each one of our the species in our taxonomic groups. And what I found, which was consistent with that previous chart, is that cnidarians, echinoderms, and decapods have a decently high coverage for a lot of their genes. I mean, some of them are still low, but overall the highest number of coverage for all 5 of the genes are in these 3 groups. And then annelids, crustaceans, gastropods, and insects also had a lower proportion. With insects really not even having any markers available for 12S or 16S. It's very low amount of markers available. But I wanted to look at the coverage of each one too. So I looked, and so if you look across this far right bar is almost consistently higher in almost all of the groups. So that's good. That's our CO1 marker. So that means that almost all of them have a really high, the highest proportion of coverage is in CO1. Nigerians also have a really high 16S, and so did Daguerreotypes. They also have a high 16S marker. And then our lowest markers were actually in our 12S, so insect hedgehog, like I said, but our 12S, which is that first bar, had a really low markers overall. And then in Decapods, 28S was also super low. So what this told us is what I wanted to target. So I don't know if we go all the way back to my beginning, talking about how I do PCR and I amplify a targeted region. Well, I want to know I was looking at what genes would be the best to get us the best coverage in our species. And that let me decide on CO1 and 18S, they're both relatively high. So that's what I was using this data, one of the ways I was using this data. And this goes back to like, we're only as good as our reference libraries. The better the reference libraries, the better resolution we can have within our taxa. The good news is that they are improving every day. They're being added to almost every day. You can go to GenBank and search them. They'll usually, there'll be an increase in reference libraries. But what still needs work is we still have those gaps in the taxonomic coverage, those annelids that we were talking about, those gastropods. Like we still have a lot of species that don't have the markers necessary. And our goal is more high quality more high quality reference sequences. And part of that is adding those ourselves as well. To help really bridge the gap that in our genetic reference libraries and help us really get to a spot where we can go in and identify almost everything that could potentially be there. And to do that, the Smithsonian is holding an informal BioBlitz in June, where we will be looking at and collecting species for reference library curation. So we'll be going out collecting and identifying these species and then creating our own barcodes to then put on NCBI as well as our Smithsonian websites. And gold. So then we have more reference libraries available, especially for the species that don't have any. And then I'm also going to be incorporating some real world data. So I'm very excited about this. I've just gotten some data back from our June sampling where I have taxonomic identification of these sequences, those ASVs. So what I'm going to be doing next is looking at what the taxonomic resolution of our real world samples looks like. Are we getting a lot that are just identified as annelid? Are we getting a lot of species resolution? And then also seeing if the species we are getting are the Andes species of interest. So then seeing if I can then get a genetic reference, genetic AMBI index going to see if our genetic work is similar to what Holly does and it's the same results. That's all I have for everybody today. I'm happy to take questions or talk more about eDNA if you guys want. I don't know if online there's questions or not, but I'm happy to give questions or talk more about my research.
Jessica, how do you know whether the reference library is good? As you said, it's only as good as the person who identified the organism. So
yeah, that's a great question. So one thing I do like to look at is who identified. So if I'm looking through the references, if I'm going to use it, I want to see who identified it. Usually when you go to NCBI and bold, you can say like, you have to put in your name. So if I put something on bold, it's identified to me. So like our Smithsonian Environmental Research Center, they put a lot of stuff on NCBI and bold. And then also one thing I love to do is I'll take, if there's multiple sequences available, like let's say there's 20 CO1 markers for one species. I'll take all of them and I'll actually put them in a tree, a phylogenetic tree. And if they were all exactly the same, it would look exactly the same. They'd have the same branchings in that tree. So then the ones that are exactly the same, I have more of a confidence in saying, yes, this is probably that species versus if there's one that's like super long and out there, not grouping with the others. I'm like, I probably don't want to use that one. You can also look at it, you can download the data. I mean, it would take a lot of time to download reference libraries for 1,188 species. But you can look at them and look at what the actual FASTA sequences or what the actual DNA sequences look like, if they're full of, you know, DNAs, A's, T's, C's, and G's. Anybody, you do have Jurassic Park fans in here? Right. So yeah, I always think about that Mr. DNA, but if there's a lot of N's in your sequence, those stand for not assigned. So if your sequence is riddled with a ton of N's, and 85% of the 85 is high, but if like 50% of your sequence is just N's, I wouldn't trust it. I would want something a little higher quality with more defined bases. You want to do what you can.
Yeah, I might be sort of missing something here, but
No.
the markers, they all have S on them. Did that refer to like set rates or ribosomes? So they're looking at transcripts, you know, or you know at that?
So those are
Why the S?
at the end, like the 18S, 28S, like 18S, 12S, 28S.
Yeah.
Those are ribosomal ITS
Right,
region
okay,
genes.
that's what I
Yeah.
thought. So in the case of insects, then, I mean, like Drosophila and, you know, all the bugs flying around here, those markers are all identified, right? So, and they're in the library.
Mhm.
So pretty much everything that we SWAT, you know, will be in the library. The question is that, you know, there are a lot of armored, right? So that's kind of the issue.
Yeah, so a lot of them, that's one thing, there's a big taxonomic bias and a lot of reference libraries, so really studied organisms, insects sometimes, but a lot of mammals, fish, bird species, they have a ton of reference, and people have been doing this and have been looking at these genes for a long time. So a lot of them will have them in their reference library, but then go back to understudied, such as Benthic, they're not regularly known, like a lot of people are like, what's an amphipod? Small precision. So that's where the issue comes, is that we don't have those references available yet. But hopefully we will. The more people study it, and inverts are cool. The more people who study them, the more genetic work we get done, the more we'll add to it.
But unfortunately, how often would you say you come across things that aren't referenced at all versus things that are?
A lot. So we know I've been looking, there are a ton of like eukaryota, and so like I don't have exact numbers yet because I just started looking, but I would say at around 40%, 40-50% usually is in what I get around that amount of things that are just eukaryota or even just unassigned, which means they don't know if they don't know what it is yet. It hasn't. So bacteria, that's a big one that we'll get some bacteria in there, but then also like if it's a bacterial sequence, it's whole nother ball game. So yeah, there's a lot of unassigned, and that's one thing we look at when we're looking at biodiversity. Just what's unassigned, what can be assigned, like what really is that diversity if it's just unassigned. It's an issue, but we're working on it. Like I said, they're improving every day.
Does that mean your name will be on their libraries for lots of species, because you'll be the first one to enter them in the database?
Well, I think mine along with a lot of
Yeah.
other Smithsonian people will be on there. But yeah, hopefully if I'm submitting them, it'll be submitted by me, and then everybody on that project will be listed as identifiers on there. So hopefully by the end of the BioBlitz, when we get all our data back, we'll have a ton of new sequences in there.
Or would your strategy be to put the expert's name on so that people would have more confidence in
Well,
the future? The true taxonomists that they.
Oh, they'll be on the author list, yeah. So basically the author list in NCBI, because I'm most familiar with NCBI, it'll say like submitted by, so you'll know like whoever submits it'll be on there, but the authors will be on there. So yeah, anybody who helps identify, we can put on there as like an author, so then they can say, okay, yeah, this is like, this person knows amphipods, so we can be really confident that this is that species, or more confident.
How will algae overgrowth affect your findings? Can your findings be used to measure the health of the
So we're benthic algae,
Well, yeah.
or just algae in general.
Yeah.
So we do get a lot of algae. We get a lot of algae sequences. For the MAMB index, what we're looking at is really looking at isolating our sequences for the inverts. But that's one thing is very interesting. Really good question. So with sequencing, it's not 1-to-1. You don't have one amphipod, one sequencing read. You do get a little bit. It's not exactly a 1-to-1 look. Like across the scale. But what we're hoping is that we can find a way to say like, okay, we see a ton of reads for algae, so we know there's an algae bloom going on, we see a ton of these reads for algae, so hopefully relative proportion wise, we can figure out how that would work. One of my goals is then to be able to use all of the sequencing reads to kind of assess the ecosystem as well, but we gotta start somewhere, so, yeah, but you could see a lot of, I mean, I get a lot of diatoms, I get a lot of algae, I get a lot of red algae, I get a ton of everything in these reads. I had 108,000 sequences, like ASVs in my last round, so it's a lot. But yeah, there is potential to use it for like algae, looking at algae, other things. A lot of people do phytoplankton in the ocean, in the water column, and they'll look at like surface and they don't find phytoplankton. So people are doing a lot of cool stuff with eDNA. It's got a lot of really cool potential.
Curious, you said 108,000 reads, that doesn't translate directly to species.
No, so 108,000 ASVs, so out of my reads, 108,000 things are saying this is something. We can say this is its own amplicon sequence variant, we know it's something. And then I take those ASVs and try to taxonomically identify them. So out of 108,000, and I had a bunch of sites, one of my sites had 435 ASVs unique to that site. So they could be, so yeah, but 108,000 between 4 different sites was a lot. And so, like I said, some of them are still not identified, some of them are just unknown. It'll just NCBI enrolled them those like we don't know what it is yet. But
I was curious, I know you're looking for benthic and fauna and avifauna, but if you took a water sample directly above, how would that compare to taking a sediment sample? I was just curious about that.
compare in terms of epifauna and fauna, or compare just in general?
Well, would you be able to detect your epifauna and infauna from a water sample? Or do you know, like?
In a perfect world, we'd be able to at least get the epifauna if it's right above that benthic surface. We try to get a little bit of the water from when we, because our planer goes down and grabs, it takes that, we try to get that surface water to at least a little bit of surface water. Those little pores in the video show a little layer of surface water. Compared, I'm sure we would get a lot more free swimming, so like we might get more fish DNA versus what I get in my benthic and fauna. I don't know exactly how they compare because I've only been eating more so the sediment, but I mean, we'd probably, if it was right above there, you'd probably get anything that was like close to the surface. You'd probably get something from the surface of the sediment. Going down in burrowing organisms, that's really dependent on the water sediment quality, water temperature, how much UV gets there. So UV light destroys DNA. So if there's a lot of high sunlight, shallow area, you might not get as much from like the surface. There's a lot of caveats.
I wanted to thank you for, I thought it was a delightful presentation of your laboratory materials and methods and techniques. To do the video on high speed, just get to see you shake the Erlenmeyer, clean it out, and get it on, try it, it was a nice presentation, to be able to,
Visualize.
take that from the Ekman Ridge and the Buck and pull it out and do a great job.
I appreciate that because that's why I was a little late because I didn't embed the videos right, so they weren't playing. And Brinson had to save the day and put them in. So I'm glad you liked it because I was like, well, I guess I could just talk through it. But stay tuned. We're working on the DNA extraction video too. So if you follow our Instagram, we're going to be posting it within the next couple weeks, hopefully. And that shows the next steps after I get all that sediment and whatnot. Then that shows the next steps of what we do to extract DNA. So stay tuned and hopefully we can post it soon. Takes a lot of editing to do those videos because the decantations for each sample, it takes me 45 minutes to an hour to do one. So, and that's
But your sterile technique is, is ever so important, and that's what's endlessly time consuming, with this work compared to, other field stuff and what that is. who cares, I mean, you got.
Yeah, the bleach, I love to say bleach and I became really good friends because I use a lot of bleach. I feel like the Smithsonian Marine Station is now the top buyer of this company's bleach because I have to buy every day before we go in the field, I switch out the bleach buckets too. So every day we have fresh bleach buckets to start and so it takes one day of sampling takes 1 gallon plus 1 liter of bleach just to sample. And then on top of that, we do that for 7 to 10 days, depending on weather. So if we have bad weather days, it's one of those things I go out, I change the bleach before we leave. We get there, if we can't get to our site. So it takes a lot of bleach. And then on top of that, the big bleach buckets in my lab, the 10% bleach solution I just ran everything down. Bleach and I are good friends.
Our clothes are ruined.
My shoes are, you can kind of see these ones have like bleach stains on them. I'm the worst with like my shoes. All of my shoes have little bleach stains all over them because I'm not great at like when I lift stuff out of the bucket and somehow get them everywhere. I stained one of my shirts and I was pretty bummed about it, but it was my fault because I grabbed it and I almost dropped it. So I like held, it was an Erlen Meyer flex big bleach stain, but it's just like a war mark on my shirt.
One of the things you said early on was the reason they were looking at doing the eDNA is because of the time intensive nature of doing the manual version of all this. Has anybody actually done any math to see like number of man hours, costs of sequencing and stuff like that versus paying
Somebody to pick
somebody
through.
to scope, yeah, and do all that.
So that's one thing we're working on. I've been tracking, that's how I know like how long it takes for me to do everything. So we've been tracking and trying to come up with like an average amount of man hours for calculations. And then with the sequencing as well, I've been tracking how much sequencing costs. And there's no actual comparison available yet, but it is for one person. It takes, so to go through a sample, sometimes it takes all these groups, 1 week to get through one sample, and we have 63 samples each period, so it can take a long time, but it took me 3 weeks to do all of this, from start to finish. So sequencing is a little more expensive, but it's getting cheaper every day. The better sequencing technology comes out, the cheaper it gets. So I would love to get you a little time sheet that's done.
Well, just, there's hours, but then there's also expertise and then there's, the cost of sequencing and then the post-processing once you get the sequences back.
Yeah.
Right now, because the library is not very, there's a lot of your time spent actually like
Analyzing.
the data, right? So cleaning up the data, doing the analysis, I was just curious. So, have we reached equilibrium yet?
You'll probably see a delivery of it, hopefully soon. Yeah, because I've been keeping track on like a little spreadsheet of how long, and then we have to, of course, like, I'm an expert, but some of the lab stuff that we're doing, as long as you teach it right, you can teach somebody who's not quite, they don't. Yeah, to do the extractions and the decants and everything, we can teach an intern, or we could teach somebody who has enough experience to work in a lab, but they don't have to have a PhD to be able to do that. So
there's undergrads and grad students all over the country doing that.
Right, and they're very important. We need them.
Thanks, Jeff.
Yeah, thanks.